Replication of DNA molecules first starts off at the origins of replication. In a eukaryotic chromosome, such as a human's, there are a few thousand replication origins. From there, helicase will attach to the DNA and begin untwisting and separating into the two strands at the replication fork, the area where the parent strand of DNA are being unwounded. This stage of DNA replication, the helicase is unzipping/separating the maternal and paternal strands and creating single-strand binding proteins. These are proteins that bind to the unpaired DNA strands during the DNA replication, stabilizing them and holding them apart while they serve as templates for the synthesis of complementary strands of DNA. The helicase unwinds the DNA in front of the replication fork, but overwinds the DNA behind the fork, creating strain on the DNA. Another enzyme, Polymerases, works to relieve this tension by breaking it, turning it, and rejoins it. In this next phase of DNA replication, the primase synthesizes with RNA primers using the parental DNA as a template, which means that the complementary strand of DNA is matching up with the strand that was just unzipped. Notice how the template strand is running from the 3' end to the 5' end, while the new strand is running opposite. This is so that the bases can pair up while running in two opposite directions in order to create the double helix shape. Double helix. Two anti-parallel strands of DNA. The initial nucleotide chain that is produced during DNA synthesis is actually a short stretch of RNA, not DNA. This RNA chain is called a primer and is synthesized by the enzyme primase. Primase synthesizes RNA primers using the parental DNA as a template. Primase, the enzyme that synthesizes the primer. Primer, the initial nucleotide chain that is produced during DNA synthesis that is actually a short stretch of RNA. Enzymes called DNA polymerases catalyze the synthesis of new DNA by adding nucleotides to a pre-existing chain. Most DNA polymerases require a primer and a DNA template strand, along with complementary DNA nucleotides line up. DNA polymerases. Enzymes that catalyze the synthesis of new DNA by adding nucleotides to a pre-existing chain. The two strands of DNA in a double helix are anti-parallel, meaning that they are oriented in opposite directions to each other, like a divided highway. Because of their structure, DNA polymerases can add nucleotides only to the free 3' end of a primer or growing DNA strand, never to the 5' end. Thus, a DNA strand can only elongate in the 5 to 3 direction. DNA polymer 3 remains in the replication fork on that template strand and continuously adds nucleotides to the new complementary strand as the fork progresses. The DNA strand made by this mechanism is called the leading strand. To elongate the other new strand of DNA in the mandatory 5 to 3 direction, DNA polymer 3 must work alongside other template strands in the direction away from the replication fork. The DNA strand elongating this direction is called the lagging strand. Stage of replication, the DNA polymerase 3 has no way to attach to the lagging strand due to the fact that there is no 3' end to begin replication. To solve this problem, DNA primase attaches an RNA primer onto the lagging strand to provide a 3 region for the polymerase 3 to begin the replication process in the direction away from the replication fork. Then, when the DNA polymerase 3 reaches the upstream RNA primer, it drops off, leaving a gap between the newly replicated DNA and the RNA primer. These short strands of DNA synthesized on the lagging strand are called Okazaki fragments. Next, DNA polymerase 1 removes the RNA primer, replacing the RNA with DNA, but there is still a gap. Finally, DNA ligase fills in the remaining gaps. This shows the lagging strand beginning to form what is now known the trombone model of replication. Due to the fact that the lagging strand has many more steps and enzymes associated with replication, this trombone-like structure helps orient the replication enzymes for repeated synthesis. Trombone model. The protruding structure of the lagging strands of DNA during replication, which helps orient the replication enzymes for repeated synthesis.